Our second speaker tonight, this afternoon, sorry, feels like tonight to me, is Scott Dobrin. Right? Dobrin. Fix my spelling. Who's an assistant professor of biology at the University of Maine, Presque Isle. Scott earned his BS in zoology from the University of Florida, his MS in zoology from North Carolina State University, and his PhD in neuroscience from Wake Forest University. In his laboratory, they use cellular and behavioral approaches in studying the effect of experience in a honeybee, using the century-old Golgi impregnation technique.
Golgi.
Golgi. Thank you.
No one else would know.
Yeah, everybody else knows. This is not my strong point here. Using the century-old Golgi impregnation technique, Scott's research asks how foraging experience alters the morphology of individual neurons in the honeybee brain. They also examine the functional implications of these changes by comparing performance of foragers with varying experience in a classical conditioning paradigm. Scott's work uses behavioral characterization, that's the same thing, all of which can be quickly and easily learned by interested undergraduates. Take it away.
All right. Well, thank you for inviting me and allowing me to be part of this panel and this great event. I'm going to talk to you today about this really cool creature, the honeybee. You've probably heard on news, on blog sites, or many documentaries that honeybees are in trouble. Colony collapse disorders are reality. Bees are disappearing, and we don't really know why right now. This is a big problem, both for the nation and for the state of Maine, because we use pollination services. It really relies, our agricultural system relies on this pollination service. I'm not going to talk to you about that today. Instead, I want to talk to you about the cool things this bee can do. Not in this state. Carl von Frisch, arguably the best-known beekeeper, he won the Nobel Prize for describing the waggle dance that bees use to communicate both the location and the quality of a food source to other bees in the hive. But he was also the first person to show that honeybees can see color. Believe it or not, before his experiments, it was a scientific fact that insects only saw in black and white. He used a series of gray squares on a table with one colored square to show that bees could reliably find where sugar reward was just by using that colored square. Randolph Menzel revolutionized the honeybee learning field by taking this paradigm and simply giving the choice between two different colors. By rigorously controlling the hue of those colors, you could see exactly how well bees could differentiate different colors, how close they could be for them to actually tell them apart. Other scientists started to evolve this idea with simple daisies. Bees could enter through a hole in the center of a colored dot and would learn to go to the matching color. Enter through a yellow, go to the yellow with a little sugar reward at the center there. The bees could use this paradigm to demonstrate a variety of abstract concepts, such as same and different. So for example, same. Train them on a blue dot, they know to go to the blue. See a yellow dot that they've never been trained on before, they know to go to that yellow dot, not the blue. Different, train them on yellow, they go blue. See blue for the first time, they'll go to yellow. They can even do this across types of visual cues. Train them on yellow, they go to blue, give them a horizontally oriented line, and they'll go to the different vertically oriented line. Above and below they can learn. They can even go across modalities. Meaning you give them a floral scent associated with a color cue, give them that same floral scent, they will go to the appropriate color cue. This is without verbal instructions. It's amazing. Scientists began to ask, can they use visual cues to go through more complex mazes? Not a problem at all. They started to use these cylinders for some technical reasons, and then asked, can the bees learn just generic rules? Can a bee learn every time it goes into a maze, make a right-hand turn? No problem. Can it learn to do a zigzag? Right hand, left hand, right hand, left hand. It's easy. This is amazing. They can all do this thanks to the brain. Yes, bees have brains, very complex ones actually. I was studying this area of the brain called the mushroom body. This area is required for this sort of learning. And interestingly, in the 90s, scientists found that this area of the brain would grow with foraging experience. The more a bee would go out of the colony and collect pollen and nectar in the field, predictably larger the actual mushroom body would be. So I was studying how this happened. What's the mechanism? I feed bees particular chemicals that would mimic increased activity or decreased activity in this area, and I could mimic the growth that they were showing there. So I asked what I thought was a kind of obvious question. Bigger brains, are they better though? So I took a established olfactory and odor-associated learning paradigm, and I created a visual one where the bees would be presented with a different colored light, and they would extend their tongue, their proboscis, out in response to it. So science doesn't always work out as you want it to, and I found out something different. The younger bees who expect to have smaller mushroom bodies actually did this task better than the older bees, who would expect to have more foraging experience. In addition, I found completely unanticipated results. The bees across the summer were not the same. The bees in the fall were later summer, what we call fall in the field. They're really bad at this task. They couldn't do it. They couldn't do it at all. Only really in the May and June could the bees do this at all. It was really unexpected. So when I talk about this to beekeepers, they want to know this. Do my bees recognize me? Do they love me? The answer is yes, and probably. Bees were presented with a series of faces, and they had to go to the target face. And they could do this really well. 3 trials, and they're 3/4 correct. You try to do that 3/4 of the time without instructions. It's amazing. So what can you do to help the honeybee? Hopefully now that you're... Oh, no, that's So first of all, these are not bees. I don't like hornets or wasps or yellow jackets either.
I'm serious, we're going to cut you off.
I see.
But we're going to dive right into the questions, and Scott's going to be here afterwards, so you can get him for these. Any questions for Scott? Yes, sir.
What can we do to help the honey?
I love this. We can recognize the fact that honeybees are not the predominant bee up here. There's 270 bees, native bees in the state of Maine, and bumblebees are probably the most obvious ones. Think about them as well. We can support citizen and open science. UMaine Farmington, totally unaffiliated with me, has a program called the Maine Bumblebee Atlas, which is a citizen science project, which would be really great if you're interested in helping bees. And just be conscientious about the chemicals that you're putting on your lawn. Okay. Yes.
Is it true that we shouldn't mow our lawn until the dandelions have gone?
I don't know.
I've been told that they particularly need that spring food.
The bees like dandelions a lot. I can tell you that.
So it won't hurt, right?
It won't hurt the bees. The neighbor might get mad.
Anyone else? Yes, sir.
I'm just curious why they switched from rectangular containers to square.
Yeah, that's a really good question. I totally glazed over it. It's a great question. Let's go back. My wife will be upset with me for doing that. If you look at it, there's little dots that are telling them where to go, left or right. They're right underneath. You can see them right there. You can see through into the next area. If you're in this box here, you can see right into whether that's your correct answer. Here, if you go into this box, it's much more difficult to see whether you're going into the right one. You can see that a little more here. If you're entering this box here, now you're looking at a blank wall instead of another hole there. So it's for it's a technical reason. I just said it because it matched my pictures.
Go ahead, Tamara.
How would they fare in terms of colony collapse compared to other places?
Colony collapse is one of these weird things. It's only ever been documented with beekeepers that have more than 200 colonies, which are these commercial outfits. And we don't have that many commercial beekeepers. We actually import most of our colonies. Maine is the second largest importer of bees, only to California in the entire nation. We imported 60,000 bees. This was 2 years ago I got those numbers. That's a lot. Not bees, colonies. That's a lot. And so colony collapse is one of these things where, because our winters are so hard, there's not a lot of beekeepers here. I will say that there are certainly some events where bees that go to the Maine blueberry fields and then have gone down to the Massachusetts cranberries, there has been some documented loss in that sort of transition for reasons that I can get into.
Could you talk a little bit about neonicotinoids and what role they might play in colony collapse?
Yeah.
Can you repeat that?
So the question was about neonicotinoids, which is by far the most common pesticide that's used in the United States. It's been recently, maybe not so recently, but has been banned in the European Union. And there's a lot of evidence to say that something is happening with neonicotinoids. Whether it's causing colony collapse, we don't know. But there's deficits in learning in honeybees and bumblebees. There's changes in the quality of queens that are emerging. There's overwintering differences, so how well they survive in winters, whether you're treating with these neonics or not. They're certainly not a good thing. What seems, my personal perspective is, there's not any one individual chemical that's the problem. It's just like with addiction and just like with drug abuse, you might take cocaine just fine, whatever that means. But if you mix it with another drug, you're going to be in a problem. You're going to have a problem. This is the problem that seems to be going on with pesticides. It's not any one pesticide that's a problem, but when you start mixing it, which is never going to be tested by the industries because you can't predict it, you have all these problems. So a recent paper came out that they studied a variety of different bees. They found 57 different pesticides in all these different bees, with the average of 4 different pesticides in an individual. And so this interaction effect seems to be the bigger problem. It's not just what I'm doing, it's what my neighbor's doing, and my other neighbor, and my other neighbor. A bee will forage in about a 2-mile diameter, which can cover a lot of lands.
Yeah.
So is it true that do bees, like, if they have a lot of different colors, do they like one color or another?
Yeah.
Are being told as a kid, like, if you're wearing yellow or something, there would be more?
Bees, particularly bumblebees, but bees tend to like blues and yellows. And what's interesting is that there seems to be an innate preference. That some bees, some individual bees, tend to like the blues, and some individual bees tend to like the yellows, and we don't really know why that is. It's a really good question.
Thanks, Scott.